Okay, so my name's Kevin Bender. I am a professor at UCSF, and I had the good fortune of working with Nadav, and in particular with Serena Tamura when she did her graduate work in Nadav's lab to develop this rather amazing technology in collaboration with Andrew Nelson, who couldn't join us tonight, but also says hi, and Perry Spratt, who's going to be mostly leading things tonight and has continued this work as a senior scientist at Regal Therapeutics to try and advance these sort of approaches into the clinic. So we're super excited. This stuff finally got published. This has been a long slog for us. It actually kind of started probably close to 8 years ago that we started working together. Um, with Nadav meeting another notorious person within this community, Stefan Sanders, I think on a BART ride. Nadav, you could probably tell us about how this actually got started.
Yeah, happy to. Yeah, thanks, Kevin, and echo that this is really informal, so feel free to at any time ask questions, stop us, and so forth. So on our end, we were developing a therapy for haploinsufficiency. Basically, haplo is half, insufficient is not enough. And so there's a lot of diseases, over 660 genes and diseases that actually, when you have one copy, you have two copies of a gene, but if one works and one doesn't, and now you have 50% of RNA and protein being made. And so the idea that we had at the time was you still have this existing copy that's fine. And so what if we pump up the volume on that copy and force that gene to make more? So instead of 50% doing 100%, and that could rescue diseases. And we wanted to do this first on a disease that's very quantitative, something that we can measure. And so we came up with obesity, which is an easy one. And we managed to actually fix the top two obesity genes in humans that lead to obesity if you have just one copy of this. And we have mice that were supposed to be completely obese that we made very lean and beautiful. And so when we were doing that, we are thinking, what else could we do? And I was dinner with Stefan Saunders, and on the way back, we both live in the East Bay. On the train, we started talking, and then Stefan suggested, "Why don't you go after SCN2A? I know this guy called Kevin." Who has worked a lot on this mouse model, has beautiful results on it, and also plays a trombone. And so basically, I reached out to Kevin, and that's how the project started. And Serena joined the lab around that time and was really enthusiastic to take this project on. And we're fortunate also to have Perry, who was very enthusiastic about it also. And they teamed up as a team to work on it. And I guess the rest is history. And Perry will tell about that today.
Yeah, great. Serena, do you want to say anything before I get going?
Sure. Yeah, it's so nice to be here. Hi, everyone. I'm Serena Tamura. Yeah, so I was really fortunate when I went to UCSF, started my PhD. I came from a career doing clinical research at Stanford, studying neurodevelopmental disorders, like idiopathic neurodevelopmental disorders. And I was really focused on trying to better understand the genetics of neurodevelopment. And so for me, when I learned that Nadav had been developing in his lab this really cool technology to correct gene dosage imbalance, and then Kevin Bender with the expertise in SCN2A and functional electrophysiology. This was just like a dream come true to be able to work on this for my PhD research. And just want to reiterate again, Perry and Andrew, I feel like we truly were partners in crime doing this. So it's just so special to come together as a group again, and so lovely to be a part of this community. Just wanted to say hello. Again, this is the work that I did at UCSF during my PhD. I'm now a scientist at BioMarin, but I just want to, just for my full disclosures, this work was done as during my PhD time at UCSF, where I graduated in 2021. So it's been a long time coming. I'll pass it back to you, Perry.
Great. Yeah, thanks, Serena. Yeah, so my journey to the start of this project was I was a young graduate student in Kevin Bender's lab, right as he started first talking about this gene SCN2A. And I was fortunate to get involved in some of that early work. And once this opportunity to work with Serena Nadav and Navneet, who was another key collaborator on this work, I had the good fortune of just getting to test all the wonderful stuff that was coming out of the iHeartTube lab, and it manifested in this paper, and now actually Nadav and Navneet were co-founders of a company that wanted to translate this work into therapies. And so I'm fortunate to be a part of that company now. And Kevin's actually an advisor to that company as well. So um, I'm going to be speaking in my capacity as a former member of the Bender Lab. But it is worth noting, just like Serena, I have some disclosures, which is um, I do have a financial stake in a for-profit company called Regal Therapeutics, which we're trying to develop therapies for SCN2A using the technology that we're going to be talking about in this presentation. And this presentation is about using animal and cellular models in an investigational nature, so exploring what is potentially possible. So Regal is working to develop a commercialized therapy. That's not what we're talking about today. I'll have a little bit about it at the end of what we're doing at Regal, but really this is the focus of the work that I did at UCSF. And so, um, any sort of speculation about timelines and success of future human treatments it is not necessarily reflective of the position of Regal itself. So I'm just speaking in my own personal capacity today. So I hope the four of us are familiar faces. We've been in or around you all at different FamilieSCN2A meetups. This one, I think back in 2019 in Seattle. I think that's where this image is from. And you can see a lot of the members of the team. There's uh, Stephen Sanders, who was really instrumental in getting this work started at UCSF. Uh, Kevin is somewhere in here. I just realized, Kevin, you're not labeled, but we have Serena, we have Navneet, we have myself, we have Roy, Chen Yu, who did a lot of work. Oh, there's Kevin. Thank you. And then Andrew. And this was a unique period in time where we were all working at UCSF together, getting this project off the ground. And I hope that many of you, the people on this call, I've had a chance to meet and interact. And if not, please say hi at the next FamilieSCN2A meetup next summer. So we're here today to talk about the paper that we just published. So this is actually Serena's paper. She really was the driving force behind this work. I'm going to speak about it just because it relates to what I'm currently working on. But I
Perry,
hope
I
you guys.
literally could not have done this without you. This is our paper. Don't be silly.
Well, Serena was the engine that really helped develop this technology, and everyone on this long author list contributed in their own important ways. And it's a real privilege to get to talk about the work of such a large and wonderful group of people. And I'm hoping today, I'm sure there's going to be lots of questions. I'm hoping to address one, what the key results of this paper are. We can have some discussion about what this means for the kiddos with loss of function SCN2A variants, and what this means for caregivers as well. And I can give a little bit of color about what is needed to translate these results from the academic context that this work was done into a potential therapy. So I really think that there are three key results of this paper. The first is that restoring SCN2A expression rescues the deficits caused by SCN2A haploinsufficiency, even when we rescue outside of early development in adolescence. So this is not necessarily specific to using CRISPR activation. In theory, any therapeutic approach that can upregulate SCN2A could have benefit for kiddos with SCN2A loss of function variants. And the therapeutic window is perhaps broader than we initially feared, and it may extend at least into adolescence and beyond if the results in mice are anything to go by. CRISPR activation is one such technology that could be used to increase the expression of SCN2A for a therapeutic purpose. And so it might be the foundation of future effective treatment in children with loss of function variants. And we show evidence towards this in that CRISPR activation, in addition to increasing SCN2A expression, reverses some of the deficits that we've been able to identify in SCN2A mouse models, as well as a few human cell models, including this novel seizure susceptibility finding that Kevin's lab developed after I left the lab. So there's at least some evidence that CRISPR activation therapies may be able to treat the seizure and non-seizure symptoms caused by loss of function variants. So this may be a slide that some of you have seen in the past from Kevin's presentations. Um, SCN2A disorders fall on a spectrum with different variants having different effects on the encoded sodium channel Nav1.2. There are gain of function variants. Those are the ones that increase the function of the channel. Those are associated with the neonatal epileptic encephalopathies. So these are the severe seizures that begin shortly after birth. And then there's a suite of variants that reduce the function of the channel. Um, these can be protein truncating variants where the channel, one of the two copies of nav 1.2 doesn't fold up properly and is, or even gets translated, and it results in half as much protein. And that leads to the host of symptoms associated with autism spectrum disorder, intellectual disability, and some of the kids with loss of function variants, seizures. There are loss of function missense variants, so these lead to an intact channel, but there are alterations in the channel's function that make it less excitable. Um, and those can also lead to the same ASD and intellectual disability phenotypes. And then, of course, not on this graph is there's a whole realm of mixed function where it can be a little bit of gain of function, a little bit of loss of function, which can lead to sort of intermediate or uh, unique presentations. And so there, for the gain of function um, variants, the therapeutic approach that you want to take is you want to either block the NaV 1.2 channels or reduce SCN2A expression. And while this is not ideal, because you don't end up at normal when you block the remaining NaV 1.2 channel, um, drugs like sodium channel blockers and antisense oligonucleotides can be effective in controlling the seizures. This approach doesn't work. These drugs are not effective in the loss of function variants. And in that case, we actually want to develop approaches that can increase the expression of SCN2A and increase the amount of the NaV 1.2 protein so that we can push the function of the cell back to normal. And so a lot of the work I initially did with Kevin was first understanding what happens to a neuron when it loses one copy of SCN2A. And what we learned is that SCN2A and NaV 1.2 supports key learning mechanisms within neurons. So there are these sort of, I think of these three key things that you observe in cortical neurons that are lacking the full expression of SCN2A. First is the action potentials, while they can initiate and propagate along the axon, they're sort of less powerful and less punchy. So they don't have the same oomph that a healthy action potential has. And the consequence of that is that the action potentials, well, they can spread down the axon and communicate to other neurons. Within that neuron, actually, the signals don't propagate as well back into the dendrites. And that lack of propagation impairs a very important synaptic plasticity mechanism. And so when you look at how these neurons are connected to other neurons, the connections look very immature, that they haven't had the proper signals to strengthen and encode sort of the proper relationship between cells. And we hypothesize that it's these deficits that are what lead to the intellectual challenges that children with loss of function variants face. And our hypothesis is that SCN2A expression is restoring SCN2A expression should be therapeutic. And one of the reasons why we think this isn't just a developmental issue is we did a whole suite of experiments in this paper that we published in 2019 that showed that if we knocked out SCN2A in later development, so after when the mice were sort of at an adolescent stage, we found that they initially looked like the neurons looked like healthy neurons, but they would over time change to look like haploinsufficient neurons. And so this shows that SCN2A and NaV1.2 have an important role throughout life, not just early development. And it really begs the question of if you could take a haploinsufficient neuron and increase the expression, would the reverse occur? So the first part of this paper, we wanted to, before we got into using CRISPR activation, we wanted to just ask the question of, does restoring SCN2A expression reverse the deficits that we observed after early development? So we developed this unique mouse line, and this has this cool property where the mouse is initially born as a heterozygote mouse for SCN2A. So this model is a loss of function variant. but we can use a series of special tools to turn this mouse back into a wild type mouse. So we take the mouse has initially one functional copy of SCN2A, and then we can use this molecule called Cre-recombinase to make it go back to having two healthy copies. So this is sort of your idealized rescue. This is what you would want to do in an ideal therapy. And what we did is we used this Cree trick to restore normal SCN2A expression in adolescents. And I don't want to get into the details necessarily of the technical details of what these graphs are quantifying. This is our measure of sort of action potential punchiness. And so you can see it goes down between the wild type mice in black and then the heterozygous mice in blue. The special mouse model initially looks like a heterozygote, but then we give it Cree, and then it goes back to being wild type, and the action potentials are rescued. So we get perfect rescue of the initial action potential punchiness phenotype. The dendritic excitability, again, is perfectly rescued. And then when we look at the synapses, those are rescued as well. So what this tells us is that restoring SCN2A expression in adolescent mice is sufficient to rescue the key deficits in neurons. Rescue crucially does not need to be in early development. And while we go on to look at using CRISPR activation, any approach in theory that can do this could have therapeutic potential. So I'm hoping, we're going after using CRISPR activation, but I hope that other groups seeing this paper are motivated to develop alternative approaches that could be just as, if not more effective as CRISPR-A. Okay, so deficits restored using this mouse model. Can we develop a technology that can do the same thing without having to engineer the genome of an organism to do this? So could we develop a therapy that does this for us? So I don't know what that box was. So this is the technology that the Active and Chris and Serena brought to the table that we spent many years of optimizing and evaluating. So in kiddos with with loss of function variants, you have two copies of every gene, two copies of SCN2A. One copy is the healthy copy, so has the functional encodes the functional NaV1.2. And then the other copy doesn't work. And in this case, it results in about half as much of the protein. So half as much NaV1.2, and it causes all the deficits that we just talked about. What we can do is we can use this technology called CRISPR activation, which the goal of it is to take that remaining healthy copy and boost the output of it. So it's like putting your foot on the gas on the control mechanism of the gene. So you sort of trick the cell into thinking that it has two copies of the same gene in an ideal case. So you get more gene expression off the remaining copy. That leads to more of the protein, and that protein goes into the membrane, and the neuron hopefully starts to function like a healthy neuron again. So the components of the CRISPR activation system that we use in the paper has two parts. So one is there's the effector system, that part has two parts. So we have a DCAS9 protein. And so what this is if you've heard of CRISPR genome editing, this is a variant of it that is non-editing. So we've taken this Cas9 protein that we can target anywhere in the genome, and we've blunted its scissors so that it can bind to sequences of DNA, but it can't actually edit it. And to that protein, we fuse this protein called VP64, which is a transcriptional activator. And by pairing that with a targeting sequence called a guide RNA, we can put this complex anywhere we want in the genome, as long as we follow a couple of rules. And so what we do is we target this structure to the regulatory sequences of SCN2A. And so Serena did a whole bunch of work to screen different guide RNAs and place this molecule into slightly different places in the genome. And she found a spot where you could place this DCAS9 VP64 complex where it would increase the expression of the SCN2A gene, and as a result, increase the expression of NaV1.2. So that was a lot of work. There's a whole bunch of slides that I could show about this, but we're going to breeze right through that, and we're going to talk about how we're going to deliver this to neurons. And to do that, we use a type of engineered virus called an adeno-associated virus. So these are non-pathogenic viruses that when they get into cells, they don't integrate their genome. They stably express their own DNA beside all of the human DNA, which and it can be engineered to deliver genetic cargo to neurons and other cell types. And so it's one of the best approaches we have right now to deliver different types of genes of our choice to neurons. And so we can package the genes that make up the system into either one or two AAVs, and then um, we can use that to deliver these components to cells. And so our system uses and it's worth noting this isn't just a sort of an academic experimental technology. AAVs are actually used in multiple approved gene therapies that have now been used in hundreds, if not thousands, of patients. And what we do is we even though they have a limited cargo space, so you can only fit so much stuff within a single AAV, what Serena did is she packaged the dCas9 complex into one AAV. And then a second AAV was designed that has both the guide RNA sequence and then also has a reporter that makes the neurons glow red, so we know which neurons were infected and which ones weren't. And so uh,
Hey Perry,
the
before we get there, I think there's a few really cool
sure.
questions from the group.
Oh yeah, yeah,
Uh,
yeah. Please
yeah,
butt in.
that relate to this sort of work and this slide. So Brian was asking, does CRISPR-A increase transcription from both alleles?
That's a great
And
question.
once you get it in there, how long does it last? Do you have to be dosed multiple times for this to work?
Great questions.
Yeah.
So the first question is an important one, and it's something we'll circle back to. But the answer is yes. It expresses both. It increases the expression of both copies. So as it is designed right now, it is not selective to just the healthy allele and the instead of the mutant allele. So we'll talk about the applicability to different types of SCN2A variants, but right now we're pretty confident that it should work in um, protein truncating variants. And there are it's an open question, but there are reasons why it might not be as effective in missense variants that lead to a protein that can insert into the membrane. The second question was about the duration of expression. So,
Mhm.
does it need to be redosed? The nice feature of AAVs is that, and why they're used in gene therapy is not only do they um, relatively safely introduce genes of interest into a whole host of different cell types, including neurons, they also stably express um, their payloads indefinitely. So you can get dynamics in the expression. So there's usually initially a surge where there's a lot more expression and then it dies down and plateaus to a stable level. That will depend on the exact promoter you use, how strong it is, the cell type specificity of that promoter. But in theory, once it's introduced, it should be stably expressed as long as the cell doesn't divide. And neurons are non-dividing cells that for the most part, the neurons that you are born with are the neurons that you keep for the rest of your life. So if this is an effective therapy, it could be a one-time intervention.
Yeah, and Prasanna asked a question that was addressed by Nadav, but the long and the short of it this is a technique that does boost the productivity of a good gene, and as far as we can tell, there's no real side effects toward to telling a gene to work harder via this technique. We haven't found one as of yet.
Yeah,
Um,
that's
and
correct.
then Jill asked a very hard question, which is my daughter's 22. Is the and we're seeing that this sort of effect is that we're seeing effects with CRISPR-A, with SCN2A loss of function, where there are therapeutic benefits even when administered relatively late in life in a mouse. Does that mean that this might be therapeutic for a person that's in their 20s already? Um, that's a really hard question to answer. There are a lot of things that go into that, including the question of whether or not we would want to deliver this to a 22-year-old, or probably 25, 26, 27, 30-year-old by the time we start to really think about these things. Um, I can tell you that what we've seen in mouse models, which continue to get older and older and older, is that when we have done any sort of rescue with CRISPR-A in a mouse as old as I am, we see some benefit. And so it seems like there's a situation where what happens with SCN2A, as Perry mentioned earlier, that it's a learning gene, that the learning seems to just go on pause and it never progresses. It never matures to a normal to in a sort of standard developmental protocol, but it also doesn't go away. It just sits there sort of waiting for signals to come back. And so that's actually a really enticing situation where we might have a situation where we can wake up the system if we give it a little bit of an oomph via CRISPR-A or via any other approach that rescues SCN2A function. We don't know what the ceiling is in terms of development for that, and that gives us a little bit of hope. Um, and I'll stop there and let Perry keep going.
Yeah, yeah, I echo everything you just said. We have a lot of hope, but I think it's too early to tell. I hope we can get either this approach or another approach to the point where it's so safe that we can just see if it would be helpful. Of course, with new therapies, that's an important balance of the prospect of benefit balanced out with the potential downsides of an intervention like this. Okay, so we Serena developed these components and she optimized it
Okay.
for. Oh, go ahead.
Yeah. Okay.
I think just keep on going, Perry.
Yeah. Okay. Sorry. Yeah. So AAV injections, we used injections directly into the brain of these mice to deliver the AAVs, and they were able to successfully deliver these CRISPR-A components. And they were able to both increase the expression of SCN2A. So this is we could get expression not just in heterozygotes, but we could actually push the expression beyond wild type levels, which is foreshadowing for later. And in these cells, we were able to rescue the action potentials. So the oomphness of the action potential came back. We could rescue dendritic excitability, and then we could rescue the synapses. Um, and so, oops, sorry. So all this is to say is we were able to recreate what we saw in those mice where we could perfectly rescue SCN2A. So that was very encouraging. We also wanted to get beyond mice. So this is more work that Serena did, which is she was able to take SCN2A stem cells. So these are human stem cells that were engineered to have SCN2A loss of function variants, and she was able to culture them and develop them into early stage neurons, like young developing neurons, and then we could treat them with the CRISPR-A components to see if we could rescue some of the electrophysiological deficits that these neurons have. And so actually, I forgot to include a slide here. We could also increase the expression of SCN2A, which is very important. But we found that these SCN2A neurons were less excitable. And then when we introduced the CRISPR-A components, we were able to increase that excitability. So this showed that we could get this to work not only in the mouse genome, but we could also get it to work in the human genome and in a functional capacity. Some of the really exciting work that was in this paper was actually this was all done after I left the lab, which is the obvious question is, can we show not just that we can rescue some of these neuronal excitability changes, but can we actually change a clinically relevant deficit. And so, as I'm sure you all know, a subset of individuals with SCN2A loss of function variants have seizures, and in many cases, these seizures are poorly controlled. Um, Part of this paper was showing that we have potentially a model of this in mice, which is that the SCN2A haploinsufficient mice are more sensitive to this drug called 4AP, um, which is a potassium channel blocking drug. And so we have a mechanism that explains why this is the case. I'm not going to get into that today, but when you dose the mice and you record their EEG activity, so this is so the brain waves that you can get when you put on one of those caps and look at brain activity, you get a lot of these ictal spiking events in the heterozygous mice. And this goes up as the drug comes on, and then it wears off. Um, we developed a systemic way of delivering these two viruses. So we use a special type of virus that can cross the blood-brain barrier and infect the entire brain, not just a small region where we were directly injecting in the previous figures. And when we did this, and we did the same challenge with 4AP, we found that the seizure susceptibility was reduced. So the CRISPR activation approach was protective of the seizure phenotype. And this gives us hope that a similar approach would not only improve the learning deficits that a lot of children with loss of function variants have, but also be protective against seizures and improve the quality of life in multiple ways. A very important question is what happens if we overexpress SCN2A? And this also ties into that question about how long does the rescue last? One of the things that I've learned the most from talking with caregivers is the importance of not rocking the boat and not wanting a therapy that can make things worse. And I think everyone is on this call probably has had the experience of trying a drug that was supposed to help your child and finding that it complicated things and sort of reduced stability. So we wanted to assess if we have something that is maybe more powerful than we expected, so it overexpresses SCN2A, is there any chance that we could actually make things worse? Because there's, of course, the worry of inducing seizures by having too much NaV1.2. And we were able to do this because of while the approach in heterozygous mice didn't overexpress beyond wild type levels. When we treated the wild type mice with the CRISPR activation system, we were able to get SCN2A expression beyond wild type levels. So some of them even twice as much as wild type. And so we wanted to look at the electrophysiological properties of those cells. So the good news is, just looking at the mice, they seem fine. They weren't overtly seizing, which was, I think, our first worry. And then when we actually looked at the electrophysical properties of the neurons themselves, they weren't any more excitable. So when we look between mice with different levels of SCN2A expression, we see that the punchiness of the action potentials goes up, sort of in a linear fashion. And if you project that out to what we were getting with the CRISPR activation in the wild types, where we get on average 175% of wild type levels, you would expect that the action potentials would look quite different, but they actually look the same as what the mice with 100% SCN2A expression and what they look like. We also did EEG recordings. Again, this is work that was done after I left, but what we found is that or what the Bender lab found was that challenging the mice with the proconvulsant drug didn't lead to any differential seizure susceptibility between mice that are overexpressing SCN2A due to CRISPR-A versus not. And so this was very reassuring. The reason why we think this occurs is the NaV1.2 channels slot into very defined spots in the membrane. There's a very there's an elaborate scaffold that controls the insertion of different ion channels into the membrane of neurons. And there may be a limited number of slots. And so if you have excessive NaV1.2, that the extra nav 1.2 channels may have nowhere to go. So you can sort of reduce the levels below 100% in the case of haploinsufficiency. But if you have to push it beyond that, um, as long as they're healthy channels and don't have a gain of function variant, the neurons don't become hyperexcitable. The consequence, though, of this is that uh, CRISPR activation might only be effective in protein truncating variants and not the loss of function variants. We haven't assessed this. I want to emphasize that this is just a hypothesis for what's occurring. But if this hypothesis is correct, it's if you have a loss of function variant that could take up one of these spots because we're upregulating both copies of the gene, in effect, it would cancel out. So this is something that is an open question. The hope is, of course, that it would be just effective with the loss of function missense variants, but that's something we can't say for sure right now. Kevin, is there anything you want to add here? Because again, this is actually more work that was done after I left.
No, I mean, a lot of I will say we've been working on this a long time, and a lot of what was challenging for us and also challenging for just getting a paper published, which is a very academic thing, is arguing with people that are called reviewers. We found out who one reviewer was, who was very temperamental in making sure that things were as safe as possible. And so there were a couple things. One of which is this, is to ask the question, if you overdrive the system, what happens? And so a lot of you are probably thinking, well, wait a minute, isn't gain of function overdriving the system? And yes, it is, but in a very different way. That's not more channels. It's channels that have different functions. The individual channels have been altered. Here we're saying if we put in extra channels, they actually don't get into the slots on the membrane that are responsible, that are required for a normal neuron's activity. And so that's something that we're really excited about, because this just says that there's a safety factor built into SCN2A therapeutics. As long as you can upregulate a good copy, um, you stand a chance for benefit. And so that's something that I think we're all very excited about. And it doesn't matter whether CRISPR-A is the way it works. If you can find another way of doing that, then great. We're all for it. And so that's what I think is the most exciting part about this result. The other thing that we did was a bunch of what's called genetic screens and RNA sequencing, and making sure that when you upregulate SCN2A, it doesn't do anything else that you wouldn't necessarily predict. And to the best of our ability, we didn't see anything untoward here. We didn't see any adverse effects of upregulating, and it continues to work throughout life. And that was some of the questions that were being asked on the chat. Um, so yeah, that's my
Great.
color commentary here.
Yeah,
Okay.
thanks for. Yeah,
Hi.
I can just add for the obesity, we didn't go that long, too long, I think here. The obesity also for 9 months, we have mice that look perfectly normal, even though they should be extremely obese.
Kevin,
I'm not sure who's asking a question. Yes, God, what is it?
it's me, Ahmed. Hi, how are you?
Oh, hey.
So, yeah, thank you for this amazing presentation so far. I'm just, I have a specific comment regarding the loss of function, because there are several actually mutations with loss of function criteria which decrease the expression of the channel rather than it changing the conductance. So maybe the system actually can be promising in these loss of function mutations, right?
Correct. Yeah.
Yeah,
That is right. I think the concerns that we have are regarding proteins that the protein is expressed, but the channel is non-functional.
yeah, yeah. So there's a class of these that actually Perry and Roy Ben-Shalom characterized very early on. One example is R937C or R937H. These are cases where a channel gets to the membrane, but it actually cannot flux any ions. It doesn't function as a sodium channel. And so that's a case where you have one good copy and one bad copy. And both of them, if we put CRISPR-A in that case, what we would predict is that both of them would be overexpressed and nothing would change, unfortunately. It's really this sort of approach would probably work best for the cases where in your gene DX diagnosis there would be an X involved. Um, so or a frame shift or something like that. That's where we know where we have very good confidence that this sort of approach would work. But it also suggests that we need to develop other approaches for other indications.
But do you think there are also some missense variants which decrease the expression on the membrane?
Yeah, I think that's a good point. Like if there's a variant that prevents the channel from properly trafficking, and we were able to show
Hmm.
that, in theory, this approach should work in that case.
Hmm.
So
Can
that's a very good point.
I ask Perry for you just to repeat what you just said for everyone in layman's terms?
Sure. Yeah.
The variant types for loss of function that could possibly work for this treatment. Um, Definitely protein truncating, which looks like a letter, a number, and then an X, right? That's what it looks like on the report. And then what about splice sites, frame shifts, and missense?
So frame shift and splice site, I think often those lead to effective protein truncation. It will depend on the exact variant. Um, and like what is if the shift leads to a premature termination of the amino acid sequence. Same for the frame shift. But you could I'm not familiar with specific cases or specific frame shift variants. But you could have, in theory, there could be ones that still lead to a protein that is able to fold up and go into the membrane. But I think for most of those mutations, this approach would apply. The comment around um, uh, protein or variants where the protein is able to form, but it's not able to traffic to the right spot. Proteins have components in them that enable them to function. So there's the ion channel part of Nav 1.2, but there are also components that tell it to go to the right spot in the cell. And if there are variants that affect the ability for the sodium channel to go to the right place, potentially this concern we have about a limited number of slots wouldn't hold because those proteins wouldn't be able to take up those slots. And so the slots would remain empty until we boost the expression of the healthy copy. Um, so in theory, it would work in those cases. But
Awesome.
I do want to emphasize that like the work that we've done is sort of the purest form of protein truncation. So this mouse model doesn't express anything off of the unhealthy allele, which is not the case for most SCN2A variants. Most of those will be protein truncating or missense mutations that change the channel function.
Thanks, Perry.
No problem. So that's at a high level the paper. There's a ton more work that went into it to address all those tricky reviewer questions that Kevin was uh, alluded to, and a lot of the hard work that Serena did to characterize the system. So if you are interested, I hope that this could be a nice primer to dig into the rest of the paper, which is as Leah said, available on Nature. And of course, if there are questions after this talk and you want to reach out to any of us, we're more than happy to take those questions after the talk, or now. This has sort of come up. The
Sorry
limitations
to
of
interrupt. I have to run to another dinner meeting, but I'm going to be on the phone with my headphones and listening, but no video.
Wonderful.
Apologize.
Okay. No,
Thanks,
thanks, Nadav.
Nadav.
Bye, Nadav.
I'm still going to be on, but just
Okay.
without a video.
Okay, so yeah, these are the key results. We've discussed several of the limitations through the questions. So one, this has not been assessed in non-protein truncating loss of function missense variants. So as one of the first questions was, CRISPR can't at this point distinguish between the healthy and the missense variant. If there's a limited capacity for Nav 1.2 2 in the neuronal membrane. Upregulating the missense variant might cancel out the upregulation. So it's uh, more technology development will be needed to find a way to specifically upregulate the healthy copy and not the unhealthy copy. You can maybe even imagine a situation where you could suppress the missense variant. But that's right now in the realm of speculation and possibility as opposed to something we can actually apply. Um, the CRISPR activation system using this paper is sort of a prototype system that has a lot of additional components that you would not want in a human therapy. So it's not ready to directly be used safely in humans, and I'll talk a bit about what's required for that in the next slide. So a therapeutic system would need to be developed and optimized into a single AAV vector with neuronal specificity and the ability to manufacture at a large scale for this to be translatable to humans. And related to that is we did some preliminary assessment of the safety, and we really hadn't no indication that the system was toxic to neurons or bad for the mice. Uh, the biotech and pharma industry has a much higher standard for rigorously testing the safety of a system. And so a lot of work will need to be done to once we have an optimized vector to identify both a safe and an effective dose before it can be used in humans. So what is needed to turn this into a therapy? That was my question at the end of this project back in 2020. And so I have been on a journey for 5 years now, learning about how do you take basic science advances and bring them into the clinic. And I'm happy to say that I'm trying to do the same with the work that was started by Serena and Nadav at Regal. And at a very high level, these are some of the things that you need to do once you have a prototype system. So one is optimizing the vector, the system to fit in a single vector. And ideally only express that in neurons. We don't want to be expressing and tinkering with cells that don't need to be tinkered with. We want to only be delivering one AAV and not two to limit the potential for toxicity and lower the dose that's required. We need to develop scalable manufacturing processes. This is actually a particularly challenging problem that people devote whole careers to, which is like, how do you create clinical grade, pure, high quality AAV in a way that is scalable to dose hundreds of patients. You need orders of magnitude more AAV to be effective in a human than you do in a mouse. And then you need to perform comprehensive proof of concept studies, like similar to the ones that we did that show that the therapy can work, but you also want to do it in a way where you're controlling for the dose, you're controlling for the amount of Cas9 that's expressed. You want to really understand what parts of the system are needed to create efficacy, and at what point can you cross up the line to generating toxicity. And everything is a toxic at some point. And so you need to identify at what level your product becomes toxic and design this and optimize the system so that there's a large gap between where you first see efficacy and when you later and where you eventually see toxicity. And that's particularly important for gene therapy because it's a one-time treatment. It's not something that you can easily ramp up like you can with other more traditional pharmaceutical drugs. It's much more like a surgery where you do a one-time intervention. And so we want to make sure when we're dosing the first human patients that we do so at a level where we're sure it will work and where we're confident that it will be safe. And then lastly is we need to demonstrate before you can market the product and give it widely, you need to show that it is effective in actual patients. And so you need to establish clinical trial endpoints that are meaningful to patients and caregivers, and also robust, quantifiable, and valid in the eyes of the FDA. And so that is a huge thing. You can go into a trial with a drug that works well, but if you don't design those endpoints right, you easily could get results that result in the product not being approved and not being made widely available. So a lot of the work that is being done through FamilieSCN2A for the clinical trial readiness study, the Dragonfly study, all the data that the families are sharing with these databases is very helpful in helping companies like the one I now work at um, establish clinical trial endpoints. So once we have a product that we can actually show that it works and get it out in the world. And so I only have a couple slides on Regal. Regal Therapeutics is a company that was founded by Nadav, Navneet, and a few other co-founders who are actually work out of Boston and Cambridge. So Jordan Dimenstein is another one of the co-founders. And what we're working to do is we in a commercial context call this approach targeted epi editing. That's a fancy term to refer to all the same things that we just talked about for the past hour, which is we're developing a platform where we can express the dCas9 module and the guide RNA in a cell type specific manner and get it all into a single AAV that we can tune the expression to the optimal level of to the optimal efficacious and safe level. We believe that by taking this approach where we have a single vector that we deliver to the specific tissues of interest, so we'd be dosing directly into the brain, that we can and reducing the expression only to the neurons that need to be in the cells that need to be intervened in, that we can safely upregulate the gene and create an effective therapy. So we're trying to develop a system with multiple layers of safety and specificity so that we can have a large safety margin between the efficacious dose and the toxic dose. And we're applying that to SCN2A. So we're actually going after two different sodium channelopathies. I'm sure many people on this call have heard of Dravet syndrome, which is very similar in terms of mechanism to SCN2A haploinsufficiency. It's caused by loss of function mutations in SCN1A. And so we've adapted the approach to a product that we're calling RT101, and we're developing that for Dravet syndrome. And then our next program is what we're developing for SCN2A haploinsufficiency using all the same tools and models that we just presented on now and are in the paper. And they're at various stages of development. They're both preclinical. We're further along with RT101. We're in the stage where we're really rigorously identifying the safety of this product. And then in RT102, we're a little bit earlier adapting all the work that we did at UCSF into a single package that we hope will be hot on the heels of RT101 and in the clinic as soon as possible. So that's all I'm going to talk about for Regal today. I'm hoping in the future there'll be a lot more to talk about in terms of our progress on RT102. I want to conclude with we've made a lot of comments about how this was a sort of a long time coming getting this paper published, but really actually if you reflect on the past decade, since Serena and I first got involved in this project, there's been an explosion of SCN2A research and the path to effective therapies is accelerating. A decade, I'm sure, is an eternity for a caregiver of a child with an SCN2A related disorder, but really in terms of how science advances, it's what's occurring is remarkable. Yeah, we're starting to see so for the gain of function kiddos, CRISPR activation is a potentially viable approach for the loss of function kiddos. And like we said, the key thing I think really to take away from this paper is even if it's not CRISPR activation, any other approach that is able to upregulate the gene, and maybe someone can come up with an effective way to target the healthy copy, and not the mutant allele. As the technology progresses, we know that this is now a potentially treatable condition, and I think that's probably the most important thing that comes out of this paper. This work involves a collaboration of many labs across many generations of scientists in this lab and a tremendous amount of investment in basic research. These are just some photos that Kevin sent me of sort of the original UCSF team when Leah first came to visit, I think back in 2016, maybe 2017, with everyone who's on this call, Serena, Nadav, Kevin, and myself. We've been at a bunch of the different Families SCN2A conferences. This is us, I believe, back in Seattle. This is the lab when I left it in 2020, and this is the lab today, or at least pretty recently. And we've had a ton of collaborators, of course, the Sanders lab, the AccuTube lab, but a whole host of other UCSF labs. A point of particular pride of this paper is actually my fiance was in the PAS lab who helped out with some of the early EEG experiments. So it was also a pleasure to get to co-publish with my partner. So yeah, we're at time, but we can turn it over to any questions that people have now. I see it in the chat that the number keeps going up, so I'm sure there's lots of questions.
Thank you so much, Perry. That was wonderful. We do have a lot of chatter in the chat, mostly about specific variants. So if any of the scientists on the call would like to answer those, that would be amazing.
Sure. Yeah. I'm having a little bit of trouble accessing the comments.
Well,
So if
I would
someone else
say,
could read them out.
yeah, I mean, a lot of the particular variants, I think that those are discussions with both the genetic counselor and with members of the Family Foundation, um, to try and get a sense of which type of variant you're dealing with. Um, I don't know whether or not we should cover that right now.
Yeah, I think beyond what we've said, it addressing every individual case, yeah, I think that is probably best.
It's okay. You don't have to say
Yeah.
whether they're eligible for a trial that hasn't
Yeah.
even started yet. They're just wondering what kind of variant they have. Is it
Oh, I see.
a
Yeah. Well,
missense or protein truncation?
in that case, because I would certainly recommend talking uh, to a genetic counselor or talking
Yeah.
to the source that gave you the test. They should be able to provide you with that information.
We're so early in the pipeline that we don't actually look at these type of mutations as much as you all actually
Yeah.
might, or that the internet might be able to verify for you. So, um,
So Mara asks a question: is there a place other than Simon's research to list potential candidates for studies? Um,
Oh, this is being like cell lines? Do you think that's what the
I'm not quite sure. Mara, could you expand on what you were asking there?
Sorry.
No, it's
Um, I,
fine.
I was trying to word it nice. My husband typed it for me. So currently we are registered under the Simon Research Foundation.
Mm-hmm,
Like put all my son's information in there. And I was just
mm-hmm.
wondering, like, with what you guys are doing, is there another place, any other places besides that to put our kids' information in that can be stored somewhere and we can be notified of these things if there is upcoming clinical trials and like our information is in there for like researchers like you to see already.
Yeah.
Honestly, I would say the best thing would be to be associated with the Family Foundation and all of the groups that are working within this domain. The goal is to work as closely with the Family Foundations as possible to ensure that the registry is as up to date there, and we work with families that way. Yeah.
Yeah.
So,
I'll say on the Regal side, the first place we go when we start really designing the clinical trial will be the Families Foundation. So um, talk to Leah, talk to other parents about where what systems have been put in place to capture that data,
yeah.
and we'll use FamilieSCN2A as sort of our go-to to get access to it.
Mara, check the chat. We put a bunch of
Yeah.
links in there
Yeah.
to access our website and all of the opportunities to get involved.
Nicole is asking a question. What's your expected timeline, Perry? Is this going to be right after Christmas?
Um, I that's unfortunately something I can't comment on, and frankly, it would be entirely speculative at this point. So we're working as hard as we can. It's uh, yeah, it's a challenging time, I'll say, in biotech right now, but everyone at our company is extremely motivated to uh, be working on this project. We've had some parents actually come speak to us, and I remember Sean Egan came. If any of you have met Sean, and there wasn't a dry eye in the room, so everyone's extremely motivated.
Yeah. Melissa asks, do we have any quantitative or qualitative measures of what function could look like when rescued? Leah's asking, do you mean cells or do you mean people? I'm assuming you mean people. I'm assuming you mean what are your kiddos going to be looking like? If I were designing the trial, and I can comment on this because I'm not the one designing the trial, but here's what I would do. What I would do is I would focus on a kiddo that actually has some seizure problems right now, because that's the easiest, most quantifiable, and most easily FDA approvable metric to assess. And we can see whether or not that metric has been rescued. Now, in addition to that, there's going to be what are called secondary endpoints. There's going to be whether they can tie their shoes, how much language do they have, whether the coordination has improved. These sort of things can be assessed, but you're right, you need something that is quantifiable and unbiased. And so the best way to start is within an individual, get an EEG, perform a treatment, and then measure the EEG as they go through the therapeutic window. And so that's what I would think an expert like Perry would do, but he can't talk about that, so he's not going to.
Not yet, at least. I hope in the not too distant future. But
Yeah. There are advantages of being an academic. I could just, as you all know, I say whatever comes to my mind. Okay. If there are other questions that people would want to just ask uh, to us directly or in chat, feel free.
I'm also happy to collect questions via email and get them over to these guys and
Yeah, yeah,
gals.
we'd be happy to, anything that we can help with, happy to help. Um,
This was such an
yeah,
honor to
yeah.
have you all here with us tonight. Thank you so, so much, Serena, Perry, Kevin, Nadav. This was an incredible treat. Any last questions before we say goodnight? Everyone's just saying thank you.
oh,
Yeah,
well,
appreciate it.
it's always the honor is all ours.
Mhm.
It really is getting the chance to actually work on projects like this that matter to people is it's a rare privilege. So thanks for the
Well
invitation,
said.
Leah.
One question, this thing is recorded, right? So will it be provided later, Leah?
Yes. I'm
Okay.
going to edit out Kevin and then I will play it. I'm just kidding.
You're going to play the trombone?
Yes, by tomorrow. I'll have it ready for you tomorrow.
Okay, thank you.
That's
Yeah,
a
Leah,
lot
if
of
you could
editing
also
you're gonna
export
have to
the comments, that'd be
do.
potentially
Yeah,
helpful.
yeah. Brian asked a question, for clinical trials, do you need primate models?
Yes, the
Do
answer is I guess by models,
you need to do
no,
safety
You need
yeah,
studies?
primates are typically used for safety studies.
Yeah,
Yeah.
but you don't need to make an SCN2A primate
No.
monkey. That actually is not necessary. It's really testing the safety of the approach, and that's true for every therapeutic that is of this genre. Any sort of genetic therapy has to be tested for safety in that respect.
Yeah.
Yeah.
That's a good question.
And will clinical trials include candidates outside of America, and will they start when they start, like Australia? I have no idea. It's
That I can also safely say I have no idea.
yeah,
But yeah,
yeah. And it's entirely possible that it wouldn't even start in the US.
it is anything is possible on that front at this
Yeah,
moment.
no. I would love to include Canada.
I personally would love to. If there are any
As a
Canadians on the call,
Canadian.
I actually am Canadian, so
Yeah.
yes. All right.
Wonderful. Well, thank you again, and everyone have a wonderful night. We'll post this tomorrow. Thank you.
Thanks
Yeah,
very
thanks
much.
everyone.
Have a good night,
Bye.
everybody.
Thank you, everyone. Good night.